Bachelor of Science Nursing Microbiology Unit 4 Antigen and Antibody Reactions. The immune system protects the body against infections by recognizing antigens and responding with antibodies. The interaction between these two is crucial for immune defense and is the basis for various diagnostic tests and immunological responses. Antigen, Ag, definition. An antigen is any substance that stimulates an immune response by triggering the production of antibodies. Characteristics, usually a protein or polysaccharide, e.g. bacteria, viruses, toxins. Can be foreign, pathogens, or self-antigens in autoimmune diseases. Contains epitopes, specific sites recognized by antibodies. Found on microbial surfaces, capsules, flagella, viral coats, or as toxins. Examples of antigens: bacteria, e.g. lipopolysaccharides of gram-negative bacteria. Viruses, e.g. spike proteins of coronavirus. Toxins, e.g. tetanus toxin. Antibody, immunoglobulin, e.g. definition. Antibodies are Y-shaped proteins produced by B lymphocytes, plasma cells, that recognize and bind to specific antigens. Structure, composed of 2 heavy chains and 2 light chains. Has a variable region, Fab, for antigen binding and a constant region, Fc, for immune function. All the 5 types of immunoglobulins details are given below. Antigen-antibody reactions. Definition. Antigen-antibody reactions are highly specific immune responses that occur when an antibody binds to its corresponding antigen. These reactions help in immune defense, disease diagnosis, and immunotherapy. The study of these interactions is crucial in microbiology, immunology, and clinical diagnostics. Types of antigen-antibody reactions. Precipitation. Reaction. Definition. A precipitation reaction occurs when soluble antigen combines with soluble antibody in an appropriate proportion to form an insoluble antigen-antibody complex. Precipitate. Mechanism. Antigen and antibody mix in a liquid medium. At an optimal ratio, cross-linking occurs between antigen and antibody. This results in the formation of visible precipitates that settle out of solution. Types of precipitation. Reactions. Ring test. A layer of antigen solution is carefully placed over a layer of antibody solution in a test tube. A white precipitate ring forms at the junction of the 2 layers. Example: Ascolize test for anthrax detection. Flocculation. Test, the antigen-antibody reaction forms fine floccules, tiny clumps, in liquid media. Example: VDRL test for syphilis. Radial immunodiffusion, Mancini's test. The antigen diffuses into an antibody-containing agar gel, forming a precipitation ring. Used to measure immunoglobulin levels in blood. Auchterlonie double diffusion test. Both antigen and antibody diffuse in an agar gel and form precipitation lines where they meet. Used for fungal antigen detection. Immunoelectrophoresis. Combines electrophoresis and precipitation to separate antigens in a sample and identify specific proteins. Used for diagnosing multiple myeloma, abnormal antibody production. Applications. Diagnosis of infectious diseases, e.g. syphilis, anthrax. Immunoglobulin quantification, identification of fungal and bacterial antigens. Agglutination, reaction, definition. Agglutination is the clumping of particulate antigens, such as bacteria or red blood cells, with specific antibodies. It is a more sensitive reaction than precipitation. Mechanism. Antibodies bind to multiple antigenic sites on the particles. This forms cross-links between antigens, leading to visible clumping. Types of agglutination reactions. Slide agglutination. Antigen and antibody are mixed on a glass slide, forming visible clumps. Example, Widal test for typhoid fever. Tube agglutination. Antigen-antibody reaction occurs in a test tube, showing clumping at the bottom. Example, brucellosis diagnosis using the standard tube test. Indirect passive agglutination. Antigens are artificially attached to latex beads or RBCs to enhance visibility. Example, CRP latex test detects inflammation markers. Hemagglutination involves red blood cells, RBCs, and is used for blood grouping and viral diagnostics. Example, ABO blood typing. Hemagglutination inhibition test for influenza virus detection. Coombs test detects incomplete antibodies that do not cause visible agglutination, used in Rh incompatibility, hemolytic disease of the newborn. Applications, blood typing and transfusion compatibility, diagnosis of bacterial infections, typhoid, brucellosis. Viral infection detection, influenza, rubella. Complement fixation test, CFT. Definition. The complement fixation test, CFT, is a serological test that detects the presence of antigen-antibody complexes by utilizing complement proteins. It is used for diagnosing infectious diseases, particularly viral, bacterial, and parasitic infections. Principle. The complement system consists of proteins that help in immune defense by promoting inflammation, phagocytosis, and cell lysis. When an antigen-antibody complex forms in the presence of complement proteins, the complement gets fixed, utilized. If no antigen-antibody complex is present, the complement remains free and causes hemolysis of red blood cells, RBCS. Steps of the complement fixation test. Step 1. Serum preparation. A patient's serum sample is collected and heated to 56 degrees Celsius for 30 minutes to inactivate natural complement. Step 2. Addition of antigen and complement. A known antigen is added to the serum. If the serum contains the corresponding antibody, an antigen-antibody complex forms. A standard amount of complement protein is added. Step 3. Addition of indicator system. A sheep RBC suspension coated with anti-sheep RBC antibodies is added. If the complement was already fixed in Step 2, it will not be available to lyse RBCs. If no antigen-antibody complex formed, the free complement will lyse RBCs, releasing hemoglobin, positive indicator of no immune reaction. Interpretation of results. Positive test, no hemolysis. Presence of specific antibodies. Complement is fixed to the antigen-antibody complex. No free complement remains to lyse RBCs. Negative test, hemolysis. Absence of antibodies. No antigen-antibody complex is formed. Free complement lyses RBCs, leading to red color due to hemoglobin release. Applications of complement fixation test. Diagnosis of syphilis, Wassermann test. Detection of influenza, measles, rubella. Identification of parasitic infections, malaria, trypanosomiasis. Diagnosis of fungal infections, histoplasmosis, coccidioidomycosis. Neutralization reaction, definition. This reaction occurs when antibodies neutralize the harmful effects of antigens such as toxins or viruses. Mechanism: antibodies, antitoxins, bind to bacterial toxins and inactivate them. Prevents the toxin from binding to host cells. Used to protect against diseases caused by toxin-producing bacteria. Examples: tetanus and diphtheria antitoxins neutralize bacterial toxins. Rabies immunoglobulin neutralizes the rabies virus. COVID-19 monoclonal antibodies neutralize SARS-CoV-2. Opsonization definition: Opsonization is a process in which antibodies coat the antigen, making it easier for phagocytes, macrophages, neutrophils to engulf and destroy it. Mechanism: antibodies bind to the antigen surface. The Fc region of the antibody attracts phagocytes. The antigen is engulfed and destroyed by the immune cells. Example: IgG and complement proteins opsonize Staphylococcus aureus for easier phagocytosis. Immunofluorescence test, IFT. Definition: the immunofluorescence test, IFT, is a highly sensitive technique used to detect specific antigens or antibodies using fluorescent-labeled antibodies. It is widely used in microbiology, virology, and immunology. Principal: fluorescent dyes, e.g. fluorescein isothiocyanate, rhodamine, are attached to antibodies. These labeled antibodies bind to specific antigens in the sample. When exposed to UV light or fluorescence microscopy, the antigen-antibody complexes appear bright green or red, indicating the presence of the target antigen or antibody. Types of immunofluorescence tests: direct immunofluorescence, DFA. Fluorescent-labeled antibodies directly bind to specific antigens in a patient's sample, used for rapid diagnosis of viral and bacterial infections. Example: detection of rabies virus in brain tissue, diagnosis of chlamydia trachomatis. Indirect immunofluorescence, IFA. Two-step process where an unlabeled primary antibody binds to the antigen, followed by a fluorescent-labeled secondary antibody that binds to the primary antibody. Used for detecting autoimmune diseases and infections. Example: diagnosis of systemic lupus, erythematosus, SLE, ANA test, detection of Mycoplasma pneumoniae. Procedure for immunofluorescence. Test: Step 1. Sample preparation. The specimen, tissue, blood smear, or cultured cells is fixed onto a slide. Step 2. Addition of fluorescent-labeled antibody in DFA. The fluorescent-labeled antibody is added directly. In IFA, the sample is first treated with a primary antibody, followed by a fluorescent-labeled secondary antibody. Step 3. Washing and observation. The slide is washed to remove excess antibodies. Observed under a fluorescence microscope. Applications of immunofluorescence. Test: diagnosis of rabies virus, syphilis, and chlamydia. Detection of autoimmune diseases, lupus, rheumatoid arthritis. Used in cancer research and pathology. ELISA, enzyme-linked immunosorbent assay. Definition: ELISA is a highly sensitive and specific technique used to detect antigens or antibodies in clinical and research settings using enzyme-linked antibodies. Principle: the test is based on antigen-antibody binding, where an enzyme-labeled antibody produces a color change when it reacts with a substrate. The intensity of the color is measured using a spectrophotometer, which indicates the presence and concentration of the antigen or antibody. Types of ELISA tests. Direct ELISA. Detects antigens in a sample, uses a single enzyme-labeled antibody that binds to the target antigen. Example: detection of hepatitis, B antigen HBsAg. Indirect ELISA detects antibodies in a sample, e.g. blood serum, uses an unlabeled primary antibody and a secondary enzyme-labeled antibody. Example: HIV antibody detection, sandwich, ELISA. Detects antigens using two antibodies, capture and detection antibodies. The antigen is sandwiched between the two antibodies. Example: COVID-19 antigen detection, competitive ELISA. Measures antigen concentration by competition between the sample antigen and a known antigen. Example: detection of hormone levels, e.g. cortisol, testosterone. Procedure for ELISA. Step 1. Coating the plate. A 96-well plate is coated with either the antigen for indirect ELISA or antibody for direct sandwich ELISA. Step 2. Blocking non-specific sites are blocked using bovine serum albumin BSA to prevent false positives. Step 3. Addition of sample. The patient's serum sample, antibody or antigen, is added. If present, it binds to the coated antigen or antibody. Step 4. Addition of enzyme-linked antibody. A secondary antibody linked to an enzyme, e.g. horseradish peroxidase alkaline phosphatase, is added. Step 5. Addition of substrate. The enzyme reacts with a chromogenic substrate, producing a color change. Step 6. Measurement. The intensity of color is measured using a spectrophotometer at a specific wavelength. Applications of ELISA. Diagnosis of HIV, hepatitis B, COVID-19 and dengue. Detection of autoimmune diseases and hormonal disorders. Food safety. Allergen detection in food products. Conclusion, complement fixation test, immunofluorescence test and ELISA are essential diagnostic tools in microbiology and immunology. They help in detecting infectious diseases, autoimmune conditions and hormonal imbalances, making them crucial for nursing and medical practice.